Welcome to this video on the direct LSTA assay, a cost-effective, $100 approach for single-cell type gene expression biomarker analysis. This method provides a direct way to measure transcript abundance for specific leukocyte cell types. Measuring single-cell type gene expression in peripheral blood is typically difficult because it is a mixture of cells. Existing methods like cell sorting or single-cell RNA sequencing are not practical for clinical labs. We have developed a third method that uses a ratio-based biomarker to get these results directly. Standard isolation techniques, or scRNA-seq, are labor-intensive and expensive. Our direct LSTA method eliminates the need for physical cell sorting by using an in silico algorithm, allowing us to obtain single-cell type gene expression results directly from blood samples. Our invention uses a pair of genes as a ratio-based biomarker. By analyzing bulk data, we can determine the expression of a single cell type without isolation, providing results in about 2 hours. Bulk expression data often masks the specific activity of individual cell types, making it difficult to differentiate clinical states. Current definitive methods are simply not feasible for routine hospital laboratory use. The direct LSTA method yields results that are highly consistent with the gold standard of cell sorting. By measuring the ratio of 2 informative genes, we achieve comparable accuracy to purified cell analysis. The success of our direct LSTA assay is built upon 3 fundamental pillars: identifying informative genes, using in silico cell sorting, and employing accurate gene expression quantification. Pillar 1 involves identifying monocyte informative genes. We focus on genes with expression levels at least 2.5 times higher in monocytes compared to the overall peripheral blood sample, which acts as the foundation for our algorithm. Pillar 2 uses these genes to create ratio-based biomarkers. By analyzing public datasets, we identify the genes with the least biological variation to serve as reliable denominators for our ratios. The third pillar is accurate quantification. We use dPCR to measure transcript abundance. For instance, specific gene ratios can serve as biomarkers for conditions like bacterial or viral infections. Our research across thousands of individuals has validated these ratio-based biomarkers. These examples demonstrate that the direct LSTA results closely track with purified monocyte data and show strong performance in clinical detection. The mechanism is straightforward. We replace labor-intensive physical sorting with proprietary mathematical deconvolution models, achieving single-cell resolution without specialized hardware. Beyond monocytes, we have validated the direct LSTA method for other cell types, such as granulocytes, identifying useful biomarkers that demonstrate clinical utility in detecting infections. Clinical applications include using these biomarkers to triage febrile patients in the emergency room by differentiating between bacterial and viral infections, with further applications currently in development. The direct LSTA assay is ready for use, with established reference ranges derived from prospective studies on healthy individuals, providing a clear baseline for clinical interpretation. This slide highlights real-world performance. The direct LSTA assay, targeting IFI27 and PSAP in monocytes, effectively identifies viral infections. Clinical performance in triage for COVID-19 patients demonstrates high accuracy, with an area under the curve of 0.98, as presented in ADLM poster number 6065. Our list of applications is growing. The direct LSTA method can detect various disease groups, including tuberculosis, Kawasaki disease, systemic fungal infections, and more. The process begins with blood quantification for gene expression, followed by an algorithmic classification into pyrogenic or interferon-based groups based on specific gene ratios. We have secured robust IP protection for this technology. Patents are secured or filed for our in silico cell sorting algorithm and the direct LSTA models for both monocytes and granulocytes, covering key markets including the US, China, and worldwide. Finally, we acknowledge our team members and recent milestones, including participation in the Droplet Digital PCR World 2025 event and recognition at the International Exhibition of Inventions of Geneva. This concludes our overview of the direct LSTA technology.